NS4210 Introduction to Fieldwork Bioscience, Dean Field Study Centre, Holly Farrington, Lauren Hewitt, Sophie Howells, Ellie Knight-Rolfe.
We arrived at the Dean Field Study Centre in the Forest of Dean where we would be undertaking our fieldwork. We began by participating in team building challenges. Our first challenge was to take the leap of faith. This was a particularly difficult challenge, but we were able to undertake it as a team through encouragement and communication. Our next challenge was to take on Jacob's Ladder. This activity involved working as a team to climb floating beams. It really tested our teamwork skills, and as we had to be aware of other members of our group and help them if they needed it. For the second half of the afternoon, we participated in various problem-solving activities. As a group, we worked together effectively and were able to complete many of the challenges. A few of these problems included completing a laser maze blindfolded and grabbing milk bottles using a rope and claw mechanism. In the evening, we went on an optional bat walk to the local church. We used the bat box detector set to a frequency of 45 kHz to detect bats in the surrounding area. The detector was set to 45 kHz because most British bats are audible at this frequency. We were able to identify bats on the detector through audible clicking sounds. As a group, we found this very interesting, especially as it was something we had never tried before. We also used the British Bats ID guide to identify the bats we saw and were able to see some bats, especially around the church and graveyard.
Our first project day involved freshwater biology. We were searching for freshwater invertebrates inhabiting Oakwood Brook, which is a tributary of the River Lid. The relevant applications behind the study could include stream water quality, assessing for pollution, understanding biodiversity, monitoring for change over time, assessing the area for SSSI or nature reserve status, or be a part of a management plan. The results may have many interesting parties, such as the Department of Rural Affairs, the Environment Agency, Natural England, water companies and land developers. The site at Oakwood Brook was situated between Park End and Whitemead at a stream at the bottom of a grassy bank on a dry, overcast day. We chose 3 meso habitats in the area to sample from. These were at the centre of the riverbed and the left and right river banks. We sent 2 of our group members into the stream at safe access points and directed them to the meso habitats. Using kick sampling, we directed specimens into a fine net by using the natural flow of the brook. Kick sampling is a method of collecting specimens by using a sweeping technique with the net and kicking the sediment up from the riverbed upstream to flow into the net downstream. We collected our specimens and headed back to identify them using a field guide. We used kick sampling again in the afternoon in order to take samples from riffles and pools with the aim of comparing species diversity in these 2 areas of the stream. Our results were as predicted. We found that on average there were more organisms in the riffles than the pools, with 100 organisms found in 30 cm squared in our sample from the riffle and only 79 organisms in the sample from the pool. The mean benthic density index for the riffle was 3.3, whereas the pool was 2.6. This matches the trend found by Brown and Brussock in the riffle and pool comparison article. However, on average, after collating our results with those of other groups, we found there was more species on average in the pool than the riffle, the opposite of our original prediction. Overall, on average, we found 7.5 organisms in the pool and 6.75 organisms in the riffle. This result could have been because each dataset was collected by a different group, so variation in results is to be expected. Had we done more repeats, then a different trend may have emerged. Please see the slide for our 95% confidence interval calculations. There was a difference in the benthic density between riffles and pools as the error bars overlap.
The aim of this day was to learn more about different terrestrial organisms that were inhabiting the Forest of Dean. Before we set off, we met at the garden room to check the mammal trap that had been laid the previous night. It was, however, empty. The type of trap used is a Longworth trap with a tunnel mechanism. The door closes when the mammal steps on the bar. On our journey to RSPB Nature Reserve Nag's Head, we managed to see a rare hawfinch in a tree using our binoculars. We learnt that a set of binoculars are essential for successful fieldwork, as well as how to focus them and use them correctly. Following this, we were taught about land management, such as who is responsible for the land and what happens when an SSSI application is applied for. Shortly after, we arrived at a bird hide where we were able to successfully view and identify a number of species, such as the mandarin duck and pied flycatcher. Our afternoon project involved walking a 1 kilometre transect 4 times to survey 3 different topics. On the first walkout, we measured the 1 kilometre transect at 100-metre intervals, using what we could find to mark the sections, such as twigs and branches. The second walk we spent looking for squirrel damage on trees, looking at 10 randomly chosen trees at each 100-metre section. Our results showed that 56% of trees had squirrel damage. The proportion of damaged trees concluded at 5.6 out of 10. Our final walk was looking at the biodiversity of birds in each sector. We identified 49 different species in total, with an average of 4.9 different species per 100-metre section. Lastly, walking along the transect, we selected 200 trees in total and found that the highest proportion of trees was beech at 32%. During our walk along the transect, we also found several trees were damaged at the base, indicating boar were in this particular area.
For the microbiology project day, we set up a lab in the Dean Field Study Centre garden room. We completed lab technique activities in the morning that would help us when we undertook our group project in the afternoon. First, we looked at lab hygiene. We used a UV fluorescing spray on our hands and then washed them. This was a demonstration to show how effective our hand washing was. After washing our hands, we shone a UV light on them and fluorescing patches showed the ineffectiveness of our hand washing. Places on our hands that were particularly dirty included around the fingernails, where 90% of germs are found. Next, we practiced our aseptic technique. We washed our benches with ethanol because it bursts open the cells of bacteria and kills them. We also worked within a 15 centimetre radius of the Bunsen burner flame and wore PPE and made sure our sleeves were pulled down to ensure skin cells did not shed onto the agar and contaminate the plate. We practiced our aseptic technique by mowing a lawn onto an agar plate. We used a micropipette to put 10 microlitres of a soil sample onto the agar and sterilised the glass spreader before using it to mow the soil sample into the agar. Next, we made a streak plate. First, we sterilised the loop and then attached the loop to a colony of Micrococcus luteus. We then spread the colony onto a sterile agar plate. In the afternoon, we began our group project. We asked the question, which of our 4 chosen hygiene products will create the biggest inhibition area? Our 4 chosen products are ethanol spray, hand sanitizer, washing up liquid and hand soap. We used the same method as before to mow the lawn with a soil sample and added a paper square soaked with a sample of each of the products to the plate. We produced 20 repeats and sealed the lids with parafilm. Upon returning to campus the day after the field trip, we had the opportunity to examine our microbiology results. As predicted, the lab-grade hand soap was the best antibacterial substance by far. The hand sanitizer and the ethanol disinfectant spray were extremely ineffective, and in most cases, the hand sanitizer seemed to even promote bacterial growth, the opposite of its intended purpose. The inhibition areas surrounding the hand soap was on average 3.8 centimetres large and was the clear standout. In conclusion, judging by the analysis of our data, washing your hands thoroughly with soap is still the most effective antibacterial method. These results were consistent across all 20 of our plates.